break. And even I would dare to do, to invite you to do some exercise. So during this talk, I will try to do with you 2 forward leaps and 1 backward one. 1 backward leaps, and I will also propose you to make a detour and discuss. So you see a big plan ahead of us. So with this I skip. So I start with the detour. I like to say a few words about about the Go game, which is a game which is a board game, which is an ancient game, highly complex, highly strategic, made of plenty of black and white stones, which tons of options to move those stones on the board, told to win the game. So it was initially something which was used in ancient time, and even I think nowadays, to train the mind of young people, even the older ones, to gain some knowledge about strategic thinking and complexity. So I tried there to discuss about a way to represent a space where a 2-dimensional space where you will get different points representing a move of one of the stones on this board game, right? So it's a huge amount of options because it is thought that the combinations represent something like the number of the atoms in the entire known universe. Okay, so it's a big thing, and it's a representation. It's an undrawing thing, so nothing compared to nothing real. It's just as an illustration. One can say that somehow the human brain over the last centuries managed to explore a series of moves into the board in order to win, up to the point where AI started to play into this game also. Initially trying to explore. Initially it was completely thought as being too complex, even initially when the people from Google started to make use of this data. And we get to the point where the different series of AlphaGo was there, and at the end, the exploration of all the different moves onto this board game was to the point that now any human brain cannot. I mean, there is no human brain able to beat the AI in that sense. So I take this example because for me it's a nice illustration to explore the relationship between AI and this game as a symbol on how artificial intelligence has progressed from something super rigid, super rule-based, to something where it's capable to induce some creativity, to make some learning, and then surpassing obviously some human capabilities. And so it's through that lens I like to make my presentation about how AI can help us to explore spaces. So you will see I will be talking very much about 2-dimensional spaces in the course of my talk, and in the context of innovation in terms of innovative chemistry and innovative and safe chemistry, right? So I will be referring to acute toxicity, but not in the sense like Nicole exposed us earlier today, but in the sense of how to make use of those tools to predict in an early de-risking status in the R&D processes or an industry like ours, the way of AI can support the de-risking of our chemistry. Of course, this has to do also with the overall picture about the NGRA, because we want to de-risk chemistry and to fulfill the requirement when it comes to the regulation and the next plan in terms of regulation. But the focus again is about early de-risking and de novo design. So talking about spaces in toxicological sciences, so I named some of them there. We can start with the chemical, then we can move into the in vitro biological space, then we have the in vivo biological space. I discriminate between lab animals and human because, as you know, depending on the chemicals, we can either move into this in vivo biological space in human if it is a drug, because you can test the molecule in human. In the case of agrochemicals or industrial chemicals, we don't do that. And each of those spaces have different properties, there are, among this chemical space, there are many different ones. When you think about chemical spaces, there is the one from the drugs, there is the one from agrochemicals, there is the one from industrial chemicals. And moving on into these in vitro biological spaces, you have also many of them, depending on the cell type, depending on the time of exposure of the chemical. You're going to explore different types of spaces, depending on the concentration of the chemical you're going to use, depending on the readout of the molecule, the assay you are using or the technology, you end up with a different kind of space, okay? And of course, when it comes to in vivo biological spaces, lab animal and human, of course, species, genetic variations, sex, tissues, time, big thing, dose, also big thing, and then the readout, okay? And one of the key things for me is about investigating the interplay between all those different spaces among each other and how much they are interconnected and how much they do overlap. What are the sizes of the different spaces compared to each other? How representative one space would be, for example, in terms of a space in a lab animal compared to a human one in a given target tissue, or from a chemical space toward another properties into the biological space, and so on and so forth. So that would be the guiding principle of my presentation today. So just to give you an example, as you know, Bayer is still the only company where we have still the pharma division and the crop protection division in the same roof, under the same roof. So we are able to collaborate with some in-depth collaboration together. Here, it's just a representation about the two different chemical spaces between Bayer CropScience chemistry and the one from Bayer pharma chemistry, okay? Where you do see some overlap in terms of chemical spaces, but also distinct spaces where either the pharma or the agrochemical sector are there, which has huge implications, for example, in terms of machine learning, in terms of QSAR models. One example I like to cite is about the initial model we used using to predict mutagenicity was a model that was trained with the pharma chemistry, and we say naively that we're going to use the same model for us in agrochemical, and then it fell down miserably. So we have to retrain the entire model in order to take into consideration the specificity of our chemical space. Another example I like to cite is about in vitro biological spaces. Here are different results of cell culture treated with 9,000 chemicals, each of them. So you see each dot represents a signature, the transcriptomic signatures, and the similarity, the distance between the dots represent the degree of similarity of those signatures. Among all those different cell lines, even the ones derived from the same original tissue, you will see plenty of differences. There is not that many overlaps, by the way. So during this presentation, I was saying that we're going to do two leaps, two forward leaps. So the two leaps, those are the ones moving from the chemical space to our in vivo biological space, trying to predict strong acute toxicity in the rat, oral acute toxicity, and another one which is in vitro biological space, in that case cell painting data to predict the acute toxicity as well. And the backward one would be to move back from the in vitro space using cell painting to de novo design molecules with this type of data. So quickly, very quickly, the principle of the cell painting assay, because not everyone is fully aware about the technology itself. Really, in a nutshell, this assay is something that has been developed by Anne Carpenter at the Broad Institute in Boston, in a lab, and this an high throughput assay where you start with different cell types, which can be genetically engineered or wild type cells. You can treat them or not, or you can just look at them just to evaluate some genetic modifications, and you grow them and you stain them with different fluorescent dyes. Staining different subcellular compartments, which are listed there.
David, just if you can stay close to the computer for the online.
Okay. Yeah, sure. And out of those images that you collect via microscope, you extract the different features, and those features are used to make morphological profiles, which are known to be associated with some properties of the molecule. So molecule having similar structure, similar type of activities, bioactivities, will tend to have similar morphological profiles. This is the overall principle of this technology, which has a huge momentum at the moment. Initially, this technology was used mostly in drug discovery, using the comparison using cell lines with gene underexpressed or gene overexpressed, and more recently, cell painting is being used both in the human safety front and also in the environmental safety front in small molecule safety. So the original seminal papers on the application of cell painting was done by the lab of Josh Harrell at the US EPA. And so you can do toxicity prediction, you can do MOA investigation, or dose response characterization, just to cite a few of them. So moving back to our two forward leaps. So just to illustrate and to be super clear about what is all about between this QSAR. So we start with a training set of a series of examples of molecules and with an associated property. So in that case, we label those molecules with a property with regard to acute toxicity, yes or no, for a given dose level. And then this training set was used to train a model. And then using a new molecule, which is not part of the training set, then you try to predict the outcome of the properties of this molecule. This is the classical QSAR. What we've done also, instead of using as an input file the chemical structure, we input the cell painting profile and associate the same level when it comes to the acute toxicity, and do the same, train the model. And then in that case, as an input, you don't input the structure, you input a cell painting profile of a new molecule, and then you ask the model, based on this cell painting profile, what is the outcome in terms of acute toxicity prediction, right? So this is the overall principle. So this thing was done using acute toxicity in the context of early de-risking. This idea for us was to find a way to eliminate as soon as possible what we call the bad guys in our analogs in our chemistry, in order to remove as soon as possible the strong acutely toxic molecule from our projects, right? For this, and Fabrice is around, yeah. So Fabrice, who did this work I'm presenting, did explore two kinds of situations, where a situation where the chemistry was known in the training set. So in that case, you will have a kind of overlap between the training set and the testing set. When you do these things, sorry, I should move to the other case. The other case is a novel chemistry, and in that case, you train a model, but you mimic the fact that you are outside the applicability domain. So you have a chemical space where the model is used to train and to learn, and then you test with an extra test set, which is not known in the training set, right? So in terms of results, so we did both QSAR models and morphological-based profile. In the first case, with non-chemistry, we observed that the QSAR model was outperforming comparing the cell-based assay. But it was the opposite when it was the case where the molecule was trained, the QSAR model was trained with a different applicability domain. So this is really interesting to see, because if you're in a situation where you want to go into an innovative chemistry, so something which is not part of your training set, going into the biology is a way to get rid of this chemical applicability domain and go in, because the biology is agnostic of the chemistry, like I like to say. Okay, that was the first example I wanted to give you today. So it's a paper, and I'm super glad because it was a preprint, and we just learned this morning this paper is accepted for publication in Chemical Research in Toxicology. So let's move on to the backward leaps. Our backward leap, it's a different beast in the sense that we need to play with big data, really in the sense of tens of thousands of examples. So we make use of data from the public domain, mostly from the Broad Institute again. So the cell painting data, it was a data set of 3,000 compounds tested in one cell line, U2S. And we did make use also of a large collection of transcriptomic profile from using a platform called L1000, where we had access to something like 20,000 molecules which were tested in many different cell lines. Okay, so we made use of this assay, of this type of results in the public domain to do this backward leap. So, and for this, we use GAN, so generative model, like what I was mentioning before. And this, I like to tell you about the working hypothesis which was made, and this idea was from the postdoc from the team, Oscar, who now is working with recursion. So the idea that Oscar had was to say that the cell painting profile of another expressed target in a cell line should be similar to the one of the wild type cell line, but treated with a potent and specific agonist of a target. So that was the idea. That was the hypothesis, the working hypothesis, right? And for this, Oscar did a training set. So in that case, he did associate the molecule with the cell painting effects and train this model. And actually, he took the QSAR model upside down. And instead of inputting a chemical structure like you see in a classical QSAR, he input a profile of this overexpressing target of cell painting and ask the model to design a molecule that's going to have a high probability to induce these effects in cell painting. Okay. So this is overall what we like to call the AI-guided safety by design. So in that case, you need to have plenty of examples. You have thousands of chemicals. You expose them to cell culture. You gain information about the biological responses induced by those different compounds. That could be by cell painting. That could be by gene expression. And then you can, using GAN, make the learnings between the relationship between structures and the bioactivity patterns to then use as an input either a cell painting and or a gene expression profile to ask the model to de novo design a molecule. Okay, so we did that as a proof of concept using. So the idea was to do new drugs hits. Of course, this idea is not to do. I'm a toxicologist. For me, the objective is to design molecules with an optimized safety profile. Okay, all this require, of course, that we would have the appropriate set of descriptors or the appropriate set of patterns which are correlated with adverse outcomes in vivo and to take into consideration ADME also, obviously. This work we are now, it's two postdocs, like Joanna, who started a postdoc at NURE, who did some work in the validation of the molecule which was generated by the model. So we synthesize the molecule and you are validating experimentally the molecule which have been designed by the GAN. So rapidly, so we provided the first proof of concept that it was possible to guide chemical design using cell painting and gene expression, coupled with artificial intelligence, and we can envision to de-risk compound using AI-guided de novo design, using specific in vitro bioactivity patterns related to adverse outcome, and somehow wipe some unfavorable safety profiles from our chemistry. So this is the AI safety by design. Alors, it has been already mentioned, there is a key aspect in all those things about the use of AI into the in safety assessment is all about data generation and cooperation. In the sense, cooperation is not only about cooperating with our brains, it's also cooperating with sharing data. So I do believe that we really need to go into that direction, just to to share and to gain out of the data which has been generated, especially the in vivo historical data, which are gold for me. And so I cannot resist to say a few words about the project Oasis, because so Oasis is a large consortium. It's a collaboration between SE in the US and abroad, and we aim to explore all those different spaces using hepatotoxicity as use case. So we're going to explore at least the three spaces in chemicals, in the chemical space. So the drug, the pharma and the drug space, the one from agrochemical, the one and a little bit of the industrial chemicals. We're going to explore plenty of in vitro biological spaces in terms of cell lines, in terms of technology. That could be cell painting, that could be transcriptomics, that would be proteomics, and hopefully maybe metabolomics. And we will explore in vivo biological space in the animal, so in the rat, in a 28-day study, and in human, because we will have access for the drug, access to the data from DD compounds, so the compounds which are drugs which are on the market and for which we get some information about the effects on the liver. I would like just to close to say a few words about the elephant in the room, because in my entire presentation I did let an elephant there, and I want to name it to close this presentation. So, and AI will certainly be key in that case as well. It's all about something that I think Thomas was mentioning, and also you, Nicole, is about exposure, because I didn't discuss about exposure. Exposure of the chemicals is essential piece of all this big picture, and AI should be there also to support and make some key learnings in the future. With this, just a couple of acknowledgements about all the people. So all this work I'm presenting is a huge collaboration effort with people in many different disciplines, from computer science, cell biology, toxicology, obviously, and we had the chance to be able to collaborate also with the colleagues in pharma. And I just want to name some of the people, in particular in Sacha's team in early talks. Claire, who is the one who is responsible for the implementation of the cell painting on site, and with whom we are collaborating a lot, and also all the students which have been there with us over the last 6 or 5 years. Thank you for your attention.